Okay. First of all, thank you for the invitation. I'm actually a replacement for Chris Thayer, who couldn't be here. She's off in Amsterdam talking about literature review. Today I'm going to talk to you about identifying chemicals to test for obesity, and using clues from high throughput data. So basically, just keep in mind that we're mining data. So we're mining data through this whole talk, and there's different approaches for mining data. I'm going to show you two of them. So I'm going to start you off with an introduction to what's called ToxCast and Tox21. These are big high-throughput screening efforts that we're working on as a part of as a part of a larger consortium here at NTP. And there are two approaches for prioritizing chemicals using this data that we've tried so far. We've tried a variety of them, but I'm just going to talk to you about two today. One is expert-driven tox pi ranking, and another one is called Sentinel chemical correlation. And then from there, we'll get lists. We'll get lists of chemicals. It's the same way that Amazon figures out what you want to buy or Facebook figures out what you want to read, right? They're not always right, but that's basically how the basic technology that we're using or approach that we're using. Now you got to evaluate the plausibility. Once you get a bag of chemicals that you've prioritized, does it make sense? So ToxCast and Tox21, these are complementary collaborative projects that have the goal of generating a community data resource, high throughput screening data. So estrogen receptor assays, androgen receptor assays, cell stress assays, and these are all done in high throughput format through various contractors or through collaboration. A collaboration we have here with National Center for Chemical Genomics. And the point is to generate a resource of toxicology-focused high throughput biological screening data on compounds of concern to the public health community. So this is a public resource. This is your resource to mine if you want to dig into it. So we didn't start this to identify chemicals that cause obesity. We started this because there are a lot of chemicals out in commerce that have really no data. So to point out the challenge here, there are tens of thousands of chemicals in use at significant levels for which there is little to no safety or toxicology information to evaluate the risk of adverse effects on human health from exposures. And our response to this challenge back in 2004 with NTP was a vision and roadmap for the 21st century, and that's a document you can pull off the NTP website. And subsequently, the NRC generated a report on toxicity testing in the 21st century in 2007. So in 2005, with the NTP initiative, we started a collaboration with NCGC to start screening chemicals. And then in 2007, around the time of the NRC report, EPA started screening chemicals through their program called ToxCast. And then in 2008, we formed the Tox21, what we've been calling the Tox21 community, where a bunch of federal agencies are working together to generate data. And this is just a picture of you recognize the woman on the second from the left there. Christening our robot up at NCGC that does a lot of this screening. So the original memorandum of understanding between the federal agencies to start the screening was between NHGRI and NCGC, NTP and NIHS, and the US EPA. And this was revised in 2010 to add the US FDA. And again, it's a community resource project where the data is rapidly released. And so all the data is generated through Tox21 within 6 months of coming off the robot. You can go to PubChem and find it. So what were the goals? We first started out here. Identify patterns of compound-induced biological response in order to characterize toxicity and disease pathways, facilitate cross-species extrapolation, model low-dose extrapolation. And this is the one we're going to talk about today. Prioritize chemicals, compounds for more extensive toxicological evaluation, and develop predictive models for biological response in humans. So we started off, we've had two phases so far. In phase one, EPA screened 320 compounds, 300 unique, primarily pesticide actives across a variety of assays, about 550 in total, very large number of assays. And these data are available in the ACTOR database. NCGC and NCATS via Tox21 in collaboration with NTP screened in phase one 1408 compounds. And EPA contributed 1462 to this compound set. And we ran about 140 quantitative high throughput screening assays. and representing 77 predominantly cell-based reporter assays. So these are like estrogen receptor assays, androgen receptor assays, various types of cell stress assays. And these data are all available in PubChem, and they're also available in SEBS. Phase 2 was started around 2011. EPA started screening about 700 compounds and about 700 assays. In addition, another 1,000 compounds in endocrine active assays. And these data are all available on the EPA dashboard shown here, shown below the where data is marked there. And then in phase 2 of Tox 21, the 10K compound library, which we've created through NTP, and I'll show you the list of all the, not all the compounds, but where all the compounds came from in a minute. So 10,000 compounds are screened 3 times at 15 concentrations in a variety of quantitative high throughput screening assays. Again, these are like the estrogen receptor assays, various nuclear receptor assays. List them out, just so here, nuclear receptor activation or inhibition, cellular stress response pathways, and so far we've screened about 30 assays. And all this data is available in PubChem if you just search Tox 20, in PubChem assay if you just search Tox 21. Now, what we're going to talk about today is actually mining this phase 2 data. And so just to focus on that, the older data is available. We're using the phase 2 data today. So this is where the 10K library came from. NCGC primarily contributed drugs. EPA contributed various failed drugs on the bottom there, chemicals from the endocrine disruptor screening program, various pesticides, things along those lines. And in NTP, we've contributed a sizable number of our NTP studied compounds from the bioassay, nominations, various Nicetum related, which is a validation group that we have in NTP, Nicetum reference compounds. As you can see, we get a very broad diversity. The diversity of compounds is quite grand relative to what you see from a structural standpoint, relative to what you see in a pharmaceutical company. Although their numbers are much larger, our chemical diversity is very large. So just to be clear, there's a little bit of confusion outside of the Tox 21 program of what Tox 21 is. It's a collaboration. And so you have data sets from Tox 21, phase 1, going up the side here, and these are chemicals, and then going across the top are assays. So Tox 21 data set covers a lot more chemicals than compared to ToxCast, but ToxCast covers a lot more assays. So, and we typically think of this in the group, so there's small Tox 21, which is sort of the collaboration, and then there's sort of the big Tox 21 data set. So we hear people refer to Tox 21, oftentimes they're including both ToxCast and Tox 21. So just to be clear, and as you can see here, there is an overlap in between the data sets with respect to the chemicals. So to get down to what we're trying to do here, how do we use this data, particularly the phase 2 data, to identify chemicals that pose an obesogenic or diabetogenic hazard? And this is, again, hypothesis generation. This is not risk. So we're trying to get to, can a chemical possibly cause activation of certain biological pathways that may have these phenotypes? So I told you the first approach that we're going to talk about is what's called expert-driven tox pi ranking. And so let me take you through that process. So when we started this, we initially went to literature mining and we said, okay, what biological processes are associated with obesity-related phenotypes or diabetes-related phenotypes? And we did this through mining of literature data. And so there's a resource called CoPub that's listed here. And basically we went through and you basically searched a bunch of key terms related to obesity. And what it ends up giving you is associations that it finds in the literature with biological processes which correspond to gene ontology terms. And this is a lot of these tools that are available on the web are related back to gene ontologies. And so what we ended up with is a list of biological processes that are associated with obesity. Now, what's nice is, as people talked about earlier, obesity is a very complicated disease, so is diabetes. But we can file these down to individual biological processes, then we can find experts in those individual biological processes to then go to and ask about the next step. So we're using ToxCast data here, so we have a very large number of assays. And so we have these biological processes that we want to know something about. And we go to these experts, and the experts are listed here. A couple of them are in the audience. And we say, here, here's a list of assays that we've run. Tell us what you think are the assays that are related to your specific biological process. And so we went and collected all that information, and once we got that, what we can do is we select out those assays and we build these tox pies. And what this basically is, and this is just an example of what of how you do a tox pie analysis. So each one of these slices over here is, say, representative of a given biological target or a given assay. And so we went to, say, Dr. Blumberg over there, and he said, we need, p-bar gamma should be a part of the model, part of the, one of the assays that predicts whether or not a chemical will be will cause adipocyte differentiation or will cause effects on some phenotype. And so what we do is we add a slice here. And then we go through and we add, obviously there are a number of assays. And so we take 10, 15, however many the expert says, and we build out these slices based upon the potency of the chemicals in this assay. So in this case, HPTE, we summed up all the estrogen receptor assays here. This is actually from a paper by David Reif, who was formerly at EPA. We sum up all the actives on the ER, and you notice the ER slice here is rather large, and that's because the activity against the ER across these multiple assays is quite strong. You look down at 2,4-D here, there's no ER activity. And you see an intermediate here with this chemical here. And so you add up all of these assays, and you basically take the area of the pie, and you rank the pie. That's simply it. And so what you get out of this output, this is actually from the original 309 chemicals. This is not the analysis that we did. I'm going to show you the analysis that we did in a minute. You get a tox pie ranking. And as you see, the area of the pie increases as you go up, basically the activity ranking. So this happens to be from a paper that we're working on, one of the biological processes called adipocyte differentiation. These are the pies. Or the different assays that were included in this model. And so what you do here is you go back to the data, and this is from phase 2 ToxCast data, and you start populating these pies for each chemical, and then you basically take the area of the pie and you rank it out. So the most active chemical, one of the most active chemicals here is something we're going to we'll revisit in a second here, triphenyltin. But these, so what you get from this is you get an output of the pies that have the largest area. And, oops, pardon me. And from there, what you can see here is there's a number of chemicals that come out on top. Not surprisingly, for adipocyte differentiation, you're seeing a number of chemicals that are related to, rotenone is a no mitochondrial toxicant, farglitazar is a p-par gamma activator pharmacologically, the melangosterol is a pharmaceutical related to progestin function. So these are just the top ones that come out here. But this is one approach that you can take to prioritizing chemicals. Basically, you're looking across a suite of assays using basically a pie-based approach or an area-based approach that is related to the activity of chemicals in an assay to rank out chemicals that you, based upon specific expert-driven selection of assays. So we've done that for both adipocyte differentiation and a number of other endpoints, another one being feeding behavior. And feeding behavior is interesting because feeding behavior obviously has a lot of neurological function associated with it. And there are a number of G-protein coupled receptor assays that were in the ToxCast data set. And so these were selected as a part of the assay set that we're going to then use to rank the compounds. And as you can see here, some of these p-par gamma, a number of different G-protein coupled receptors. This is a serotonin receptor, this is a dopamine receptor, and all of these, you add basically all of these up and you take the composite area based upon the activity of the different chemicals in all the slices, and what you get is a list of chemicals here. Down the bottom, these are the top 10. Not surprisingly, based upon what was selected, they're identifying a number of compounds, chlorpromazine, a serotonin receptor effector, another serotonin receptor effector, and haloperidol, all of which have been shown to be obesogenic, if you would, or cause weight gain in the human population. So in our prioritization, taking 700 compounds from ToxCast and ranking them out based upon assays that we've selected that are biologically related to the phenotype, we've identified a number of compounds in the top, probably top 8 here, that are known to be associated with obesity in humans. So this sort of proves out an approach, potentially, that we could use to go through 700 some odd compounds and take the few compounds off the top that might actually have a phenotypic effect related to obesity. So that was more of an expert-driven approach. We said, okay, here are our assays, pick the assays, and we'll identify the chemicals for you that are active in these assays. And we'll rank them out, and basically here's a set. Here's a set of ranked compounds that you can now go ahead and test in your various different systems. And we're actually following this up with a number of the investigators that were listed there on the slide. So I want to talk about another approach that I'm a bit more familiar with, which I call Sentinel chemical correlation. So one was expert-driven, the other one is going to be purely data-driven. So start you off with a premise. Chemicals that exhibit similar biological properties across high-throughput screening assays will potentially exhibit similar in vivo biological properties. And identifying chemicals with similar HTS profiles to known obesogens, so basically you're pulling out patterns using a fish that you know a lot about, if you would, will enrich for untested chemicals in the library with such properties, meaning chemicals that have obesogenic properties. So how do we do this? So we take, this is the phase 2 data, we take and we compile all the phase 2 Tox 21 data, and this is just a big spreadsheet of chemicals by assays, and these are actually activity values that we've recorded. And there's a number of different ways of recording activity values, and I'll take you through that next. But basically, this is just a massive data set that you can now run correlation on. It's really that simple. This is probably a little bit too much depth in what you need for understanding what I'm doing, but this is a typical dose response from a chemical that's been run 3 times in, say, an estrogen receptor assay in Tox 21. We take this and we first fit a curve, and from there, we identify a number of metrics. We identify first a noise threshold, basically the activity at the base of the curve. Identify a point of departure, which is basically the dose at which you start seeing activity above the noise threshold. An area under the fit curve. And then we identify a value called a weighted area under the curve that takes into account both efficacy and potency. Now this, so we've used, and we've also in this situation, we've also taken AC50s, and that's what actually you'll find in PubChem if you go there. What we're going to do today is actually take the activity value called the WAUC. And so imagine that big spreadsheet that I showed you before, but it has WAUC values instead of AC50 values. So it captures, again, it captures both efficacy and potency. So the next thing is you take this big data set and you basically run chemical correlation on it. So imagine these are the 6 chemicals that you have, right? As you can already see, some of you probably can already pick out the chemicals that are related to each other based upon biological activity. It's very similar. And so basically, what we do is we build these relationship networks on correlation. And from that, it gives rise to this. All right, so now we have a map of relational space for biological activity of the chemicals. So what we can do now is we can go in and pull out prototype chemicals, right? So one of the prototypes for adipocyte differentiation is a PPAR gamma activator called rosiglitazone. That's been used quite extensively in the human population. And so what we do is we take, this is just a way of rendering the networks, rendering the relationships. This is rosiglitazone here, and we basically go in and fish out all of its closest friends, those that have the strongest similarity patterns across the data set. And what you find, not surprisingly, is a number of chemicals that are pharmacologically related to rosiglitazone. So this is a proof, sort of an internal proof of the idea that this approach identifies, groups chemicals in a similar fashion. We've done the same thing with triphenyltin. And as you can see here, when we do it this, a lot of the triphenyltin behaves very similar to the tributyltin, and a number of other organometallics. So again, very consistent with what we understand about the biology of these chemicals. They're all showing similar patterns across this large swath of data. Now, we've built an actual browser where people can go in. If you have a chemical, you've got a Sentinel chemical that you want to look at, you have your favorite organometallic, you just start typing in up here. This happens to just be bisphenol A, and we have all sorts of correlation metrics that you can pull down. And you can basically ask, my favorite chemical, which I know is a prototype agent for causing adipocyte differentiation, I want to see what other chemicals behave very similar to it in the data set. And therefore, and then I can go on to test that in my system if I would want to. So there's another test of truth, which I'd like to just talk on. So I go fishing. I throw out that line, and I pull in a bunch of chemicals. How do I know I've got anything worth a damn? Any truth in that bag? Am I just getting random association? So when I do this, I'm going to ask, what are the characteristics of the chemical neighbors? And so really what we're doing is very similar to gene annotation enrichment analysis, but we're doing chemical annotation enrichment analysis. So imagine you have pre-annotated buckets of chemicals, right? And you basically pulled out these chemicals from your network, your correlation network, and you start populating buckets. And lo and behold, one of your buckets is disproportionately enriched. And it just happens that 4 out of 5 of the chemicals in this bucket are, in the PPAR alpha bucket, are PPAR alpha activators. So most likely the chemical that the chemical that you searched with is probably also a PPAR alpha activator, because you've pulled out a bunch of chemicals that are very similar biologically to that chemical. Those 4 chemicals that all fit in this bucket. So we had to build this because it doesn't exist. So we went to find chemical to biological annotations. And we found a bunch of chemical to receptor annotations, chemical to toxicity annotations, and a variety of different chemical to toxicity, pharmacological activity, mesh terms, and then pathways from the comparative toxicogenomics database. So imagine this is basically, we have all these buckets that we built from chemical to biological annotations. And this fellow here, Jungkook Kerr, who was actually at University of Michigan when we spoke with him, built this, worked with him. We built this interface, and you can drop in CAS numbers. So basically, you fish out your chemicals, and then you throw them in this enricher, if you would. And so it's just taking the CAS numbers. This is our list here. Drop the CAS numbers in, and you basically get out, just like you would with gene ontology enrichment analysis, you would get out a set of enriched processes or categories of biological activity for these chemicals. So just to clarify, if we have enriched for chemicals that affect a metabolic phenotype, what characteristics should their neighbors have? Assuming my hypothesis is correct. If I got a bag of chemicals that are particularly enriched, what annotated biological process should I see? I'm thinking probably fatty acid metabolism, maybe some neurological function, something thiolizidine diones, which are the PPAR gamma class of chemicals that are known to affect adipocyte differentiation. I should be pulling these chemicals out if my hypothesis is correct. So what do we pull out? These are the enriched chemical annotations, and this is actually from rosiglitazone. I went fishing with rosiglitazone, and I pulled out chemicals that have similar activity across the Tox21 assays, and I said, okay, what are they enriched for? And it turns out there's a number of different categories here that they're enriched for that make sense. And obviously not increased lipid metabolism, probably decreased lipid metabolism, but there seems to be an inversion of this phenotype that is actually associated with blood lipid metabolism. When you go actually digging into it, it's related to the few PPAR alpha activators that are in that enriched group. Thiolizidine diones, transcription of insulin responsive genes, PPAR gamma agonists. Again, all of these things make sense. If I'm fishing out something that is probably associated with a obesogenic phenotype, these are the type of things that I should probably find. So with that, let's close. Sorry. So a couple of conclusions. ToxCast and Tox21 data sets are complementary in a manner that covers a broad chemical and biological space, and it's there for you to mine. These resources are out there for you. A variety of approaches can be used. You don't need to use my approach. If you've got a better approach or the two approaches I have, please apply them and go for it. So we've used expert-driven biology-based supervised analysis. So we said, pick the assays, we'll make the pies, and give you a rank. And we've used unsupervised sentinel chemical correlation. And we go find a chemical that we know is an obesogen, We pull out the chemicals that are associated based on biological activity, and that becomes a hypothesis set. And then chemical annotation enrichment can help to evaluate the plausibility of the prioritized chemicals. And then finally, just some acknowledgments. The folks that worked the most on these studies, Ray Wah, Shi, Dan Svoboda, from the EPA, Dane Filer, David Wright from Richard Judson, and of course Chris Thayer and Dr. Bucher, who is in the audience here. And then finally, just some acknowledgments. We use a lot of different resources, a lot of resources that people rarely give them credit for, but if these didn't exist, we wouldn't be able to do what we do. Thank you.
Thanks. Just don't go away yet. Are there any clarifying technical questions people have?
Hey, Scott. Bruce Blumberg, UC Irvine. As you know, we've done a lot of validation on our testing of these kinds of predictions, and the results are not as good as we'd hope. I think at the bottom of it is the disparity between the activity in the different assays. So, for example, if you take the PPAR gamma assay, the Adagene assay that ToxCast uses, and the original NCGC assays that ToxCast use, and the NovaScreen, there's absolutely no correlation among those three. And if you now plug in the assay, the gene blazer assays that NCGC is doing for Tox21, those don't correlate either. Mm-hmm. So given a very disparate set of results on the same receptor, I wonder how that's going to affect the quality of the data that you get out from the approach which you described, which sounds very reasonable and should give good results.
Yeah, it is a significant challenge. Part of the challenge that we're dealing with is actually the lower potency environmental chemicals. What I can tell you from reviewing the Tox21 data, pharmacology works very well. The positive controls run very well. When you start getting up into the higher micromolar dosing and start looking at activity there is some variability. And we also notice there is a significant amount of variability in calling AC50s even within a given assay. So how does this affect the predictions? From the ToxPi standpoint, I think it has a very significant challenge. But based upon internal scaling within individual assays, where you actually see covariate movement of the chemicals as a function of being in a given assay, we're using the correlation analysis approach, where we take a Sentinel chemical, it actually works surprisingly well. Right. But that actually doesn't really look specifically at the individual potencies in an assay and scale them within themselves and try to compare them to reality. It just looks at a pattern, and a pattern that's scaled within each assay. It's like a rank-based sort of thing, as opposed to which then, the rank actually seems to stay together. The chemicals move up and down. So we have a couple of estrogen assays where estrogen is really weak, but so are all the other chemicals. We're not really weak, but weaker than you would expect it to be. So it moves in groups. Right. So it certainly can affect it, particularly with the ToxPi-based approach, as opposed to using all data.
Thanks. And any other questions? So I got one question for you.
Yeah, one over here.
Oh, you're hiding in the corner.
I'm hiding. I don't even know if is this on?
Yes.
Okay, so my question is kind of stemming from Bruce's comment, I guess, in that a lot of the ToxCast or Tox21 assays they're all gene expression for the most part, right? And there are no good assays for obesogenic type activity from chemicals. So there are no good assays for lipid accumulation. There are no metabolically competent assays and such. So I think that might be something that might lead to some of the disparity in the results, specifically making it more difficult to identify chemicals that may have some sort of obesogenic effect.
So we are moving, in the next generation of Tox21, we are moving to more complex emergent cellular phenotypes, things like lipid accumulation, high content screening, and we'll have those things. But you're correct, we're typically measuring the point of contact between chemical and biology, which is the receptor. And we certainly have challenges within our screening systems. They are transformed cell lines, things along those lines that don't have metabolic activation. But the next generation of assays that we'll be working on will have a lot of that sort of have those capabilities.
Okay, well, thank you very much, Scott. We'll have a panel discussion at the end here. And Dr. Baruki, you're up.